Welcome to our second video on creating a 10 to 15 minute scientific presentation. This is the second of a two-part video sequence on creating a short scientific presentation. If you recall, the first part focused on creating an introduction for that scientific presentation. And there the focus was on how to structure a brief introduction that sets the stage for the rest of a talk. In this video, we'll focus on creating the body of the 10 to 15 minute scientific presentation, design method, data results, and conclusions. And the focus here is on the overall structure of the rest of the presentation. Note that the same patterns can apply for a longer talk as well, but in that case you expand the scope and/or apply the patterns to a succession of research questions. Recall that we begin a presentation with the introductory CCQH pattern. We move from the big picture to a well-defined problem hypothesis. We start with a context. That gives us the big picture. Then we move to the complication. The complication tells us what's unknown or uncertain about the issues raised by that context. Then we have the question, either implied or explicitly stated, raised by the complication and should be answerable in a way that will resolve the complication you've raised. And then finally we have our hypothesis, and the hypothesis focuses not only on the work itself but the presentation. In effect, you're going to present the test of your hypothesis. This introductory rhetorical pattern is like a funnel. It moves from the big and general and to the narrow focus of the hypothesis that you're presenting. And recall how in part 1 we applied the CCQH pattern to create an introductory slide for an actual neuroscience experiment. Our research work explored how reducing maternal nutrition affects the brain serotonin levels of fetal baboons, and we focused on each of the components. We looked at the context, why serotonin deficiency is linked to various issues, and then what we know and don't know, the fact that MNR reduces transporter protein expression, but the actual reduction of the protein itself was not known. Then we focused on the experiment, which contained the implied question. Let's look at these levels of proteins in baboons who are subject to nutrient reduction. And the hypothesis was that yes, we would see an accompanying reduced protein expression as a result. So after the intro, we need to create the other three components of the presentation: design methods, data and results, and conclusions. And everything focuses back on the hypothesis. The hypothesis is the structural foundation of your presentation. Everything has to relate back to that hypothesis, and it's an iterative process. It kind of reflects the whole research process itself. And design methods can be divided into two components: logical and technical. Now, when you design individual slides about design methods, you need to keep your categories sharply defined. After all, you only have 10 to 15 minutes to present your information. And you need to move from broad to specific. And most important of all, you need to focus on your audience. Who is in your audience? What do they know? What do they not know? What do they need to know? And what do they not need to know? So let's look at an example based on the presentation that we started in part 1. Now here's a design and methods slide. Design methods in study of serotonin deficiency in fetal baboons when mothers are subject to nutrient reduction. First, we looked at the study population. We had a control group and a maternal nutrient reduction group. Then we established a nutrient program. And from 30 to 165 days of gestation, for our control group, we had ad libitum feeding. And for the maternal nutrient reduction group, 70% of the calories available to the control group. Then we collected tissue with general anesthesia at 165 days of gestation. Then we analyzed that tissue using immunocytochemistry with image analysis to quantify the protein expression. We looked at the fraction, the density, and examined the amount of 5-HT7R in the frontal cortex and brainstem, and looked at the TPH from the brainstem as well. And we used these statistical methods. We used the rank sum test. The data was expressed as a mean plus or minus SEM with a confidence level set at 0.05, and the CTR values presented first. Now notice on this one slide here how we encapsulated the entire design and methods of the study and categorized them appropriately. And note how this slide uses the horizontal qualities of PowerPoint. In effect, we've created what amounts to a word table rather than a succession of bullet points. On the left, you can see a kind of classic bullet point slide, but notice on the right how much easier that is to read by creating a word table. A word table takes advantages of PowerPoint's horizontal flow. Bullet points, on the other hand, make you process words vertically, which is more appropriate for prose documents than PowerPoint slides. So take a moment to look at the way these two slides are different. And as you structure your design and method slides and even your results slides, strive to create word tables rather than lists of bullet points. But the most critical thing to do is to create 2 to 3 word categories for each of your design items or method techniques. Here we have our design, here we have our methods. Study population, nutrient program, tissue collection, tissue analysis, statistical methods. Easy for an audience to follow, easy for you to present, thorough but fast. Note how this slide combines design and methods and just addresses the technical components of design. You could have another slide that would talk about the logical characteristics of your design, or you could talk to that logic. Now, your data results make up the bulk of your presentation, and make sure, of course, that they map precisely onto the methods that you use to gather data. So in this case, the expectation is going to be, we're going to look at the protein expression in the frontal cortex, comparing the control versus the MNR group. We're going to look at it in the brainstem, control versus the MNR group, and we're going to look at TPH in the brainstem, control versus the MNR group. And you're going to have your illustrations and data in that kind of parallel fashion. Because you've set up your design and methods well, your data and results are going to follow logically as well too. Avoid the hazard of getting bogged down in data discussion. It's a real trap for a lot of presenters. Keep in mind that you should devote 1 to 1.5 minutes per slide. And if you've set up your design and methods correctly, the data will flow naturally. Finally, conclusions should circle back to the hypothesis. Was the hypothesis correct? If so, why? If not, in whole or part, why not? One slide for your conclusion. Again, you only have 10 to 15 minutes to make a presentation. Now let's look at the final structure. When you do a 10 to 15 minute scientific presentation, organize with 10 to 12 slides maximum. Remember, 1 to 1.5 minutes of talking per slide. Now your exception is the intro, which will take a little bit longer to present. And that should be the outer limit, 1 to 1.5 minutes per slide. So we start with our intro, follow our CCQH pattern. A couple of minutes. Then we get into our design and methods. Then the body of our presentation, the real meat, the data and results. And then finally, our conclusions. Then we have one final slide that's really important that we hadn't talked about, and that is next steps. Because remember, all research is designed to lead to additional research. Questions lead to additional questions. And so you should conclude with where this research is going to take you and what your next steps are going to be. So here you see the big picture of a brief 10 to 15 minute scientific presentation. A highly structured introduction, laying out design and methods clearly, presenting your data and results in a compact way, coming to specific conclusions, leading to next steps. If you think about your brief scientific presentation with this overall structure, you'll create a powerful, compelling presentation for your colleagues.